Okay, so we were talking about polymorphism, and now you have a reference to a movie. Um, when we're talking about new combinations of alleles that might occur through time, a lot of time, by the way, when we're talking about evolution, um, we're actually talking about those new combinations that occur or are produced through recombination. Um, when complementary sequences or parts of greater sequences, greater molecules, greater chains are exchanged between chromosomes, meaning that one sequence is moved to another and one is moved to the first one. That is what we call creating recombinant chromosomes or recombinant DNA, meaning that a whole section from one chromosome was moved to the other and vice versa, which is important, of course. So once again, you're referred to a movie. Recombination in such a case is regarded to as crossing over of chromosomal material, meaning that the sequence that moved from, let's say, chromosome 1 is undergoing a process called crossing over to chromosome 2. If you would imagine an imaginary bridge between the two, they are crossing over that bridge to the other side. There's not really a bridge, but that's where the crossing over part comes from. So during meiosis, we have 4 copies of chromosomes that are created, right? During the bivalent phase of the cell, or in the cell, we have all 4 copies that are together, right? So every copy is called a chromatide. When we exchange the material between pairs of chromatides, and when that occurs, that exchange, we create what we call a chiasma. The process itself is called breakage and reunion. So, once again, we have 4 copies that are exchanging materials between 2 pairs, right, of chromatides, creating a chiasma. So that process by itself is called breakage and reunion because we're breaking up something and connecting it back in a different way, right? So since 2 out of 4 of the chromatides are reacting, only 50% will be recombinant, right? Once again, we're talking about pairs. So if only a part of the 2 strands and not full strands go through a crossover process, we are producing a hybrid DNA. Okay, is created. Once again, when only a part of the 2 strands of DNA cross over, not the whole, and we're not replicating the whole molecule, we are producing what we call hybrid DNA. Okay, so exactly like we have explained with chromosomes and gave the specific example of cell division, right, and replication, we're also talking about the crossover process in DNA itself, producing a hybrid DNA molecule. So now we have a hybrid DNA molecule or stretch sequence that is located between two sequences of the parental DNA. Because if you think about it, we had one sequence of the parental DNA. Now the sequence that crosses over is inserted inside, so we have one sequence of parental DNA, then the part that underwent crossover, and then another sequence that was originally there. Okay, so it's kind of simple. And you're referred to a movie, enjoy it. If there is a possibility of mismatching occurring, it's going to be during recombination. In such cases, the mismatch may actually be recurred. Okay. So going on to the middle of page 21, you have those key concepts that you can see that you're asked to read and make sure that you go over them. I think it's a good idea before you actually read the chapter. And now we're going to talk about proteins being long chains of amino acids. I truly hope and believe that this is not the first time that you've heard this. So you have heard of amino acids, and you know that proteins are long chains of amino acids, right? So now we're going into the second mechanism that is extremely important to understand. The first one was the base pairing, right? So we understood the idea of the nucleotides. We understood the idea of nucleic acids. We understood the idea that we have bases, right? 4 bases. Well, basically 5, but in DNA 4, and RNA we also use the U instead of the T. So we have the bases. We have base pairing. Those were the leading ideas until now, and we saw what happens when they are mixed and mistakes happen, right? Now we're going to understand how amino acids are represented in DNA, in RNA or /mRNA, and how does the base pairing principle is connected to amino acids. So while the nucleotides determine the structure of DNA as a molecule, simply the sequence, right, one after the other, they also have an important role in the creation of the proteins and other factors. So the sequence has a very important implication on the protein itself. So DNA codes for amino acids, in other words, and this relationship is called the genetic code. So the genetic code emphasizes that DNA, through its sequence, includes orders and information to produce amino acids that are the building block, so to speak, of proteins. So DNA codes for amino acids that are used to produce the protein. And a protein is a long chain of amino acids connected in different variations and combinations to produce that protein. Okay? But the sequence itself of DNA is made up of the nucleotides, right? And the bases and the base pairing. So the sequence of the nucleotides represent the right order of amino acids in the protein or factor that is going to be produced. If you mess up the sequence, you might, not necessarily, you might mess up the sequence of amino acids. And then you might mess up with the protein and affect its function. All right. Furthermore, a mistake that we call a mutation, right, in a gene may alter the sequence, may alter function. This is not new. We talked about this a few times, but repetition, right? So that's kind of a recap. All right. So you're referred to a movie. Enjoy the movie. Buy yourself popcorn, whatever. So some of the codes for amino acids or other factors that information has to be understood, or make some other factor or component of the process understood. So the amino acids or the codes for amino acids that are hiding in the sequence of DNA must be read by some kind of apparatus, and in this case it's going to be a protein apparatus. Okay? Since we have in DNA 2 strands, one strand is enough to actually code for the amino acids. And it means that the base pairs are not necessarily needed for the coding of amino acids. It's enough to have one strand to have the information for what amino acids are needed for the protein. Okay? Okay, so every 3 nucleotides represent a component that we call a codon. One codon represents one amino acid to be produced. So for instance, theoretically, assuming that all the codons in a sequence, in a molecule, code for protein, I'm going to say that again. Assuming that when you have a certain molecule, every 3 nucleotides that are called a codon actually code for a protein, then if the molecule has 300 nucleotides, that means that it has 100 codons, because I'm going to divide 300 by 3. Every 3 nucleotides produce 1 codon, and that means that that sequence, that molecule, includes the information for 100 amino acids that will produce 1 protein that has 100 amino acids. Okay? So, once again, 300 nucleotides theoretically mean 300 codons, that if they are all codons that code for amino acids, we'll have 100 amino acids that produces 1 protein with 100 amino acids. All right, you're referred to a movie. Enjoy it. Once again, you have this in very big in the slide. It says 1 codon equals 3 nucleotides equals 1 amino acid. Okay, so let's say that a molecule has 3,333 nucleotides. How many amino acids is the polypeptide gonna have? You can answer yourself. So we have an apparatus that has information and gets the information from DNA or mRNA, right? It has that sequence of nucleotides that every 3 make a codon that represent a certain amino acid that should be produced and added into a polypeptide, into a chain of amino acids, right? And that apparatus is able to read the codons, decipher them, and understand that this triplet, this sequence of 3 nucleotides, this codon, this triplet, is the order to produce a certain amino acid. In other words, every amino acid has a different triplet, a different sequence of 3 nucleotides that represents it. Okay? At least one triplet. Okay? And that apparatus is going to move along the gene from one end to the other, deciphering the codons and giving instructions to what amino acid should be added next in the chain. So it's like having a thread and producing a necklace, right? With some kind of apparatus reading the instructions and giving the orders to add the right bead to the necklace. Assuming that the necklace at the end will have the right order of beads exactly like the instructions should have. That's what we're doing here. Alright? So information that is non-readable, meaning the DNA, undergoes transcription to a format that is readable, meaning mRNA. The information, the codons on the mRNA are translated into amino acids that are added one to each other to produce a polypeptide. We'll go over that again and again and again, I promise you. Okay, so the reading direction is from the 5' terminus end to the 3' end terminus. The reading is thus said to be from the N terminus to the C terminus. Okay? The genetic code has a starting point, meaning that every sequence, because it has junk DNA, has a certain sign that we will learn about that tells the apparatus this is where you start reading the codons that code for amino acids. Otherwise, if it reads from the wrong place, the sequence will be wrong, the amino acids will be wrong, and we might get nothing. So we have a certain sign, a starting codon, that says to the apparatus, start deciphering the codons from here, not before and not after. This is the first amino acid that needs to be produced. Okay, and that deciphering apparatus knows to identify that sign and start deciphering from there. Okay, so it's very sophisticated, very simple, but also sophisticated. Okay. If you understand that the sequence of nucleotides determines the sequence of codons, that determines the sequence of amino acids, can we allow overlapping? No, because that will change the accurate sequence in a certain strand in the right direction, and that will result in the wrong outcome. So overlapping is not allowed. Okay? Right. Okay, so now we went from nucleotides to codons. Now we're thinking in threes, right? In triplets. But that may produce two different types of mutations. The first one is what we call that only one codon is affected, meaning that there's a base substitution within the triplet. So we have 1, 2, 3 base pairs, right, or bases in the triplet. Let's say that only this one is affected, all right, this one or this one, whatever. One of the three is affected. This will affect only this codon, right, but it doesn't affect the rest of the codons. So this is called a base substitution. But the worst part is if the sequence itself is affected, meaning that the frame shift of the triplets moves. Then we are in a greater problem, because that means that something has either been inserted or deleted from the original sequence, and now the triplets are not the right triplets to represent the right codons, to represent the right amino acids. Hence, the protein is not going to be produced accurately. So the second type is a major problem. Most likely, if we have the second type, meaning a frame shift, the protein is going to lose its function completely. Okay? Alright, let's stop here. You are referred to a movie, right? And we'll continue in the next video.